This is episode 293 of the Stem Cell Podcast, Cell Atlases, with Dr. Sarah Teichmann. Hello, everybody. This is Dr. Daylon James, also Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. If you're enjoying the listen, please rate us and leave a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Sarah Teichmann, who's from the University of Cambridge. She's on the podcast to talk about her research on single-cell genomics. We've also got our usual roundup of recent highlights and stem cell news. That's coming up in just a second.
But first, the world's best and brightest in stem cell research and regenerative medicine convene once a year for the ISSCR annual meeting, and you are invited. Join nearly 4,000 scientists in Hong Kong, June 11th to 14th. Register at isscr2025.org, and we can't wait to see you there.
All right, I'm leading off the roundup with I don't want to call it an old story, but it's kind of a review, but it's like at the same time, the history of embryonic stem cell research as well as the future. It's everything. And it's actually a twofer in this phase of the roundup. My first stories are going to be these reports on the phase 1 / phase 1/2 trial of stem cell-derived dopaminergic neurons for Parkinson's disease. And this is kind of a bit of a review, and you should look back on the episode we did at Blue Rock where we talked to the actual pioneers behind at least one of these papers. But both the stories are very much the same, and I don't need to review what the need is here. Other than to say this is, in terms of the history of the field, this is one of the first major targets that got a lot of attention and funding. It's been a lot of major progress, and it's one of the first to the clinic and to advance to this point and to have impact where there really was no treatment. So it's all of the promise and enthusiasm, excitement surrounding stem cells and therapies in two stories. And the bottom line is, of course, dopaminergic neurons, replacing them in order to stop these dyskinesias, right? And that's putting it simply, but there's plenty of history there. I would invite you to go back to any one of 20 episodes where we've discussed this at length in the roundup or with the guest. But the bottom line here is that we have two stories that are very similar in that they both recruited a handful of patients. In the Blue Rock trial, they had 12. In this other trial that came out of Japan, they had 7. And let's just review the authors of these just at the outset here. Maybe perhaps more familiar from the Blue Rock group and Memorial Sloan Kettering. We had Vivian Tabar leading this, along with Lauren Studer and Claire Henchcliffe, who were leading this one story from Blue Rock. And then we had Ryosuke Takahashi and Jun Takahashi, who were both from Kyoto University, associated closely with the CIRA there. No relation there, I don't think, on those two lead investigators here. So the studies were very much the same in that they targeted this, the bilateral treatment and into the putamen of these cohorts, and they showed safety and tolerability, very minor adverse events that were mostly associated with either the injection, transplant, or the immunosuppression, but nothing necessarily related just to the cells. Nothing observed that was related to cells at all. So that's really good news. And they went out to a year in both these studies to look at safety and tolerability. I should mention in the Kyoto study, actually, they were looking out to 24 months as a kind of secondary endpoint, and in the Blue Rock study, they were looking out to 18 months. And that's where I think the more juicy details in these studies are in terms of the differences and similarities. One more thing to mention in terms of the approach is that there was a low and high dose in the Blue Rock approach. And also, very significantly, while that was this Y cell H9 line, the kind of founder ES cell line, In the Japanese trial, they used an iPS cell line, but it was allogeneic. So they had this founder iPS cell line, which by design was the most common Japanese haplotype, right? So it makes sense that they have this founder line. But this was clearly a departure from the original vision of using, iPS cells from each individual patient, which I think a lot of lessons from that eye trial, kind of changed the approach here. But at least a proof in principle here of those two approaches, iPS versus, traditional ESL, blastocyst-derived ES cells. In terms of the outcomes, as I said, safety and tolerability, I would encourage everyone to have a look at the paper because there is some subtlety here. But I would say overall, my take was it seemed like both were effective in stalling, I guess you would say. There wasn't a advancing of the symptoms and the dyskinesia or the pathology overtly at least. But I would say the Blue Rock showed a more impressive result. And the fact that there was a low and a high dose, I think there also showed that like the number of cells really did make a significant difference here in terms of the outcome and the symptoms in these patients. Um, and like in terms of proving the points here, yes, they reached their primary outcome. I think the really encouraging thing here, where the secondary outcomes, and most notably, no graft-induced dyskinesias, which is key because in the history of using fetal grafts, that was a major problem, and also retention of the grafts. They both use the methods to show that these grafts are retained out to these ongoing time points. And what's so great about these studies is that we're going to be looking at these patients because it's not one of these, compassionate use cases for the heart xenotransplants as a counter example. That these patients are going to be around for a while for us to follow the outcome, and not only like how the disease progresses, but more importantly, if there can be some reversal of symptoms, which I think there's encouraging signs. Although notably, it should be mentioned that there was no cessation of the drug treatment. These patients in both trials stayed on their pharmacokinetic program for reducing dyskinesias. And in the Japanese trial, actually, that led to some increase in the degree of dyskinesia as the symptoms worsened with the progression of the disease. So a lot of details there to look at in terms of parsing out which of these therapies had which effect or what you can glean from looking at the results. But for me, the most encouraging thing is, of course, this looks like a success with more results to come. And it's been done in such a careful way. And as we were talking about before the show, these results being paired in the same journal, although they were submitted, looking at the metrics, they were submitted and accepted around the same time scale. But them coming out at once, I think, is such a boost for all of us, and also a demonstration of the measured approach that we're taking towards this exciting future of cell therapy.
Yeah, if I had to summarize both of these studies in one word, it's promising because that's what we're getting at here. These are early phase trials, the results of early phase trials, as you alluded to, phase one, two, and you did a fantastic job recapping both. A lot of parallels here. Obviously, the pluripotent stem cell approach unites both of them in terms of the generation of the dopaminergic cells. You got the iPS on the Japanese side, and of course, the ES derived cells on the Blue Rock side. But both trials show promising results for safety. That's what you want to do in these early phase trials. And that's enough of a green light to progress towards later trials. And the really exciting things, like you alluded to, those high dose receiving patients, and how some of those higher doses alleviated some of the symptoms or potentially stalled the progression of the disease. So that opens the door for the next higher phase of the trial, two, three, etc., which is inevitably going on right now in both locations around the world. This really represents a tremendous milestone in the field. It's really, I think, a gold standard for how pluripotent stem cell derived trials should be conducted. Like you said, rigorous science is the foundation for both these trials. We've chatted with Vivian DeBar, we've chatted with the Lorenz, of course. This is founded on decades and decades of incredible basic science, and hopefully it's going to lead to a fruitful therapy for patients who are desperately in need of one.
Absolutely. And fun fact here, I always get a bit of joy out of the fact that my very first scientific paper, this is 30 years ago after my freshman year in college, in Bob Burke's group at Columbia College of Physicians and Surgeons, had Claire Henchcliffe as a co-author. So her and I have been working together now going on 3 decades, and it was indeed on this Parkinson's disease model. But clearly she's outpaced me in her progress in the work, and this is a measure of how long everyone has been working on this. And also, just one final note here that I think is really important, and I don't want to kind of score which of these is a more effective, like, is it better to use IPS or Y cell or whatever. I think that this is all early days for all these studies, and there's been a lot of progress and there's a long way to go. But I would invite everyone to listen to that Blue Rock episode, because what really hits you there is that they have to create a whole new paradigm in order to deliver these cells. Like, they're inventing the instrumentation and the approach, combining, high resolution imaging and precision instruments, and inventing it, building the plane while it's in the air. I shouldn't use that analogy, because again, the rigor there and the care they've taken, these are the human patients, human lives. But I want to think that the experience of Tabar's group and really being primary movers in this surgically may account for some of the better results that maybe were uh, and I don't want to put too much on it. It's a small n, but the low dose, high dose is a lot to it. But I think the message there is that as we move forward and people get better with this, and there's more adoptees of the surgical methodologies and improvements thereof, the sky really feels like the limit. And I never thought that the brain, of all places, would be this vanguard of, like, not only experimental and ambitious therapy, but highly effective and curative. So this is so great, Arun. I love it for a million reasons.
Yeah, and one thing that we highlighted on that Blue Rock episode that I think is also worth highlighting here is the courage of the patients who actually received these therapies, because this
Absolutely.
was incredibly, this was invasive, right? This is really going into the brain. This is uh, really tricky stuff to do, and so it takes a lot of courage on the part of the patients and their families to actually subject themselves to a cutting edge trial nonetheless, but anytime you're dealing with the brain, you got to be extra careful of the results and the outcomes here. So thanks to the patients, thanks to, of course, the folks at Blue Rock for sharing their work with us a while back. We'll have to grab a beer with Stefan Arion at ISSCR in a couple months in celebration of these papers, this paper. So great stuff here. Moving on to another Nature paper, actually coming from someone else who is probably going to be at ISSCR and has been recognized by ISSCR multiple times over the years, and that's Takanori Takebe from Cincinnati Children's. Of course, Dr. Takebe has been in the organoid field for many years now, developing really cutting edge liver organoids, being to be able to model various forms of liver disease, um, with also an eye towards the translational side of things. The first couple stories that we covered, of course, were inherently translational. They're results of clinical trials, but the organoids that they're developing in the Takebe lab could also potentially have clinical implications down the road. That's not the primary focus of this study. This is more like a tool generation study, how they've actually been able to make these multi-zonal liver organoids from human pluripotent stem cells. Okay. And this is again another Nature paper. I don't know if it was actually, yes, it was in the same edition of Nature as the prior clinical trial results. So hepatocytes, right? These liver cells that we all know about, there are a lot of different types of them. There's not just one size fits all for hepatocytes. There's a lot of different subpopulations of hepatocytes, and they're segregated spatially across the liver, across the portal central axis of the liver, and are important to understanding and maintaining homeostasis in the liver. And also, of course, they are critical for injury response too. And there's a ton of biomolecules that are associated with hepatocytes in the liver, most notably ascorbate, bilirubin, for example, bioactive molecules, right? They've been described as having really a critical role in directing zonal fates in the liver. And that's what this story is about. It's about making these organoids, liver organoids, more complex by incorporating these zones that you would normally find in the liver in vivo. And so until now, we haven't been able to recapitulate that in vitro, that zonal liver architecture. And that's exactly what they did here in the Takebe lab. First author here is Hassan Al Reza. Um, Side note, and we've talked about this before on the show. I think if I had to pick one place in the world where organoid biology is really at the forefront, I would pick Custom, the Center for Organoid Biology at Cincinnati Children's. I mean, they're just doing unbelievable work here, and this is just another example of that. So they showed that preconditioning the hepatocyte-like cells derived from pluripotent stem cells, um, actually were able to exhibit that induced zone-specific functions associated with urea cycle, glutathione synthesis, glutamate synthesis, all stuff that you would find in vivo, right? And they, like I said, they developed this zone-specific liver organoid actually by co-culturing ascorbate and bilirubin, those two molecules that I mentioned earlier, hepatic progenitors derived from human iPS. Okay. So after they did the co-culture, after they did the analysis of what these things are actually producing and the zonation of these organoids, they did, of course, the single cell, which is what you got to do for any new organoid tech or organoid differentiation protocol. The single cell showed and then confirmed that indeed you got the zonal patterning that you're finding in vivo as well. It actually identified a hepatoblast differentiation trajectory that dictates the different subtypes of hepatocytes that I alluded to: periportal, interzonal, pericentral human hepatocytes. And again, this is all human iPS derived, importantly. They took it a little bit deeper, did some more epigenetic analysis, and showed that the zonal identity of these hepatocytes is orchestrated specifically by the ascorbate or bilirubin dependent binding of EP300, this particular protein, to either TET1 or something that we're familiar with, HIF1 alpha. Okay, HIF1 alpha has a lot of implications in hypoxia signaling, but that's not exactly the context they're looking at here. And then the clinical implications of this. And this is just a segment of the paper towards the end. They did the transplantation. They did the transplantation of the self-assembled, zonally patterned human organoids, the liver organoids, and actually showed that it improved the survival of immunodeficient rats that actually had a bile duct ligation. So they had like a liver injury model in these rats. And it was able to alleviate the symptoms associated with hyperanemia, like this hyperaccumulation of ammonia, as well as hyperbilirubinemia. So a lot of a bilirubin accumulation, which are hallmarks of certain forms of liver disease. So just by putting the organoids in there, you're able to cut down on those phenotypes in the rats. So a lot of promise here. Multi-zonal organoid system coming from the Takebe group. It's I think the focus of the study is it's used as an in vitro model system for modeling liver disease and potentially developing new therapies for liver disease by establishing this scalable, screenable, advanced system that has the multiple zones that you would actually find in the liver. But then, of course, you got the clinical implications as well, where they showed that once you actually put these new liver organoids, zonal liver organoids into a rat system, is able to alleviate some forms of liver disease. So it's got both elements, and probably that's why it's a Nature paper.
This is so cool. Like most of the work that comes out of Takeda's lab, it's just imaginative, and this is like, I guess, an idea that we've held out ever since people started around mini livers, right? And the real clinical impact there being that it's a modular thing, right? Where you could theoretically make these little tiny livers en masse and deliver them, and they could function and do the job of the whole liver. So yeah, it sounds great, but it's been so hard to recapitulate that. And I think it's taken a real stroke of imagination that isn't a surprise. This is a bit of an aside, and I don't want to give too much fire to this because it aggravates me that this kind of happened in a way. And I hope that Dr. Takebe does not take offense. But he always lives in my mind for this Ig Nobel Prize, which yeah, he ended up getting the Ig Nobel for enteric breathing, which people call butt breathing, of course, because it's Ig Nobel, Ig Nobel. But I remember this during COVID, and people were dying. I think he had a person, someone in his, either related or somehow who died, and like that's how he leveraged his imagination to come up with something so out of the box. And it annoys me that people don't recognize. I mean, he started a company with it, and it's like actually legit and could be very important with or without a shortage of ventilators. Like it's an important advance that got, I think, a little shade because of the Ig Nobel. But I think, and I bring it up just because it underscores, I think, the imagination that Takanori is running with on all fronts, like at all times. And when you see him at the meeting, you see him at ISSCR and he's super measured and chill and speaking very quietly. Everyone's leaning in to here's what we've got to say. But his brain is firing all the time. So this is another example of that. Very impressive work. Can't wait to see what comes next.
I agree with you. I think he's probably one of the most unique and creative minds that we have in modern stem cell biology. I know he has, and I think part of that comes from his background. Like he has a very artistic background. He's really interested in design and architecture, which is kind of exactly what he's doing here with the production of these organoids. You're kind of custom designing the architecture of these multi-zonal organoids to make them do what they're supposed to do. So, yep, shout out to the Takebe Group, another Hallmark paper in a string of Hallmark papers that's been coming out of their lab for the last like 10 years plus.
Absolutely. This is a unrelated. It's a cell story, but it's really super hack. I love this hack because it shows again with the stretch of the imagination how you can apply cells sort of beyond their physiological function. And this is a story in the brain. We've been talking a bit about the brain. We led with the brain, and the takeaway there, which we talked a bit about, is how inaccessible it is, right? Both developmentally, understanding it, the pathology of neuropsychiatric disease, all that, but also just like how do you treat disease in the brain? A big part of the obstacle there is the blood-brain barrier, which is a good thing, right? We want to keep a privileged environment around the brain. And the blood-brain barrier does that by regulating the influx, efflux of molecules, peptides, cells, even uh, to from the central nervous system. But also from a therapeutic standpoint, it's a huge impediment to delivery, right? And there's been a lot of approaches to try and get around this, right? You can like use carriers type for antibodies, biologics. They've used these carriers, but in order to get them to an effective level at the brain, it's like 2% of what there is evident in the plasma. So if it's effective in the brain, you get 50 times that level in the plasma. And of course, if there's a target there that's in the periphery or non-central nervous system organs, right? We talk about this all the time. Off-target effect is going to be killer, not safe. You can also deliver right to the brain, right? There's direct injection, uh, mechanical delivery, right? But sometimes it's either multiple treatments that are required that's invasive and painful, uh, diminishes efficacy a bit, or you can have like a neurotoxicity or immune response in the case of like AAV, for example, adeno-associated viral therapies have elicited this neurotoxicity that's really limited their application in the central nervous system. Also, let's go, right? Talking about cells, you know where I'm going with this, but they've tried that with cell therapies using NSC, so neural stem cells. And in the preclinical studies, as well as like some clinical studies where NSCs have been transplanted into actual patient brains, there's like the tumorigenic potential has been a little bit troubling. So we need something. We need something else. And that's where the lab of Matthew Blurton Jones comes in. And this also senior author here on this stem cell story is Robert C. Spitale. Sorry if that's a mispronunciation. Both these guys are from University of California, Irvine. And previously in the lab in the Blurton Jones lab, they developed this protocol for making human-induced pluripotent stem cells into microglia, right? So why am I talking about microglia? Because as an alternative to NSCs, which could cause tumors, microglia maybe are a good call, right? You can inject them. So they've been injected in these cells that they made from iPS cells. You can also get microglia from hematopoietic progenitors. Um, and you get them from iPS by way of a hematopoietic progenitor. But when they inject those cells into a xeno, doing a xenotransplant in immunocompromised mice, they mature into microglia in the brain, but they're homeostatic. They just chill out. And they act like human microglia in terms of their phenotype. And here's the key point: like, there's no evidence out there that microglia generate tumors. There's no like tumor or microglial origin that has been described, right? So this sounds like a really good candidate, and that's what they went with, this group, and they really went extra on the proof of principle here. But first, just in terms of the approach, because I think it's really important, they edited these cells that they could ultimately make into microglia using CRISPR to express this enzyme neprilysin that degrades these alpha beta plaques, right? Or it's an alpha beta degrading enzyme that degrades these plaques. But here's the key: that neprilysin is under the control of a plaque responsive promoter, which is CD9. Okay. And what does that even mean, plaque responsive promoter? And I'm going to go into this because I think it's really key in terms of how modular this approach can be. They just identified this was a key part of the study, is they looked in and they said, what do these microglia do in response to plaque? Well, they looked by the expression, they saw that CD9 goes up. So they hooked up their circuit that they're going to make to CD9, because that's a key element, is that you want the responsivity of this neprilysin to be activated in the context of pathology and not healthy tissue. Again, we always talk about it, off target. So that's a key element to this hack, which I'm going to elaborate on a second. But that's the first step, right? So they have this neprilysin under control of CD9, which is a plaque responsive. And then they do this other kind of separate hack that they published earlier, where they engineer these cells so they're immune to this PXL, some chemical, right? So they can treat the mice with the chemical, and the cells then engraft into the whole brain, right? So they're able to look in two ways. They can treat the mice and they get engraftment into the whole brain, or they can treat the mice and they get a local engraftment. So they have these two kind of paradigms of engraftment. But the key here is they like ramp up the degree of brain-wide engraftment. Then they test this whole thing, the neprilysin guy, in these Alzheimer's disease mice, and they find that it does a great job, right? It reduces a lot of measures of the pathology. But the key observation here that's notable is that like the real phenotypic rescue in terms of reduction of plaque load, in terms of like reduction of the dystrophic neurites, and astrogliosis, preservation of neural density, they showed only when they had full bore microglia, brain wide microglia engraftment. So I think that was a key observation in this proof of principle that shows that maybe the scope or limitations, even you could think of it as, but what you need to do in order to have therapeutic efficacy. And then they just go even more. I don't think they needed to do this to get into cell stem cell, but they did it anyway. These reviews must have been tough. They went into two separate models. One was cancer, so breast cancer metastasis to the brain. And the other demyelination, which, modeling multiple sclerosis. So like two other complete like classes of disease. And here's the key, going back to that initial approach. They show that the microglia can respond to those pathologies. So they can identify, therefore, another gene to incorporate into their circuit, not CD9 in that case that's plaque responsive, but they get something that's cancer responsive or something that's demyelination responsive. Point being is that this is something that you could really adapt to whatever approach or whatever disease that you're interested in the central nervous system. Of course, it takes a bit of a lift to design the circuit and feedback and response elements, all that stuff. but I think this approach as like a modular approach is world class, and targeting a compartment that has been really challenging, and is the seat of a lot of diseases, not surprisingly, that are fatal and untreatable.
Yeah, this is a really cool study. I think in part because of what you alluded to at the end there, the modularity of the system, the adaptability, and how you could really just use this as a delivery modality, right? You have the microglia. They focused on the nephrolizin here as a proof of principle, but you can imagine how future studies might incorporate other payloads, and they alluded to some of this, like CAR, chimeric antigen receptors, neurotrophin, chemokines, because you can target anything, like what you're alluding to in the brain potentially, like in situations of other neurodegenerative diseases, like Alzheimer's stuff, like tau, alpha synuclein, TDP-43. These are all open targets for a delivery modality like this. So I think it's really promising. They got to think about other modalities, other potentially less invasive methods of delivery to look at as well, and figuring out the dosing, because this is way preclinical. Of course, this is in vivo mouse model, whatever animal models. But if you want to turn this into an in vivo clinical application, you got to start thinking about some of these dosing strategies and all that. But I'm sure they'll get there. This is fantastic work coming from my neighbor, actually, Matthew Blurton Jones, just down the road, about 15 minutes as the crow flies from me, from my house here in Irvine. Shout out to UC Irvine. We actually had Eileen Anderson on the show about a year ago, and they are rapidly growing as a powerhouse in biomedical research down here in Southern California, and are really at the forefront of neuroscience right now.
Yeah, Claire Hinchcliffe. Just they got her on board there as well just recently. So it's a big show over there in the neural space. But yeah, man, the limitations here, I think, are clear in terms of, yeah, way preclinical. A lot you got to do. But it's interesting that we just take for granted that you have brain-wide engraftment of a human iPS-derived hematopoietic stem cell turned microglia. Like that's just like the foundation of this story. But I think, as you said, also, yeah, the tool set here for discovery, there is the idea of applying this clinically, and that I think is the main thrust, of course, right? They're targeting these three conditions that are the basis of disease. But just think of how some clever scientists are going to apply these tools, which are like established now, right? They have the microglia that are targeted to enable this ramped up engraftment. Um, I can imagine there's going to be a lot of discovery from this as well that then can perhaps be leveraged in a way that is more amenable to a therapeutic paradigm that's safe and effective. But I love a hackaroon, and this is the hackathon in this one paper.
Hackathon, certainly. I mean, we're fans of tools here on the show, brand new, exciting tools. And indeed, we're going to wrap things up with one of the coolest new tools in stem cell biology. And that's these blood vessel organoids. That's become all the rage these days. People are taking these blood vessel organoids and fusing them into all sorts of other tissues. My lab actually has some cool collaborative work going on actually with UC Irvine in this exact area. So shout out to Irvine in more than one approach, more than one area. This is coming from Barbara Troutline's lab, and this is a very collaborative effort, as you might expect, from not just Dr. Troutline, but also Joe Penny, as you have mentioned him previously on the podcast, Joseph Penninger, as well as Jay Gray Camp. Really a trifecta of incredible stem cell biologists and computational biologists as well. And given their various expertise, you might figure out what this story is. This is basically an atlas of the developing human blood vessel organoid. Very collaborative effort that was just published in Cell. First author here is Marina Nikolova. We've talked with Dr. Penninger a while back, and more during the COVID days, because he was adapting some of his approaches to study COVID-19 and the mechanisms of COVID infection, SARS-CoV-2 infection in endothelial cells and the vasculature in particular. But that's really one of his major areas of focus. And indeed, they're developing these human blood vessel organoids. It's a really cool system to model human vascular development and disease. You're an endothelial guy. You can appreciate this, of course. And then Barbara Troutline, who is one of the rising stars in computational biology and computational stem cell biology. They leveraged her group's expertise to use single cell multiomics in combination with genetic and signaling pathway perturbations to pretty much reconstruct the development of the human blood vessel organoid. And this is relevant. Of course, our guest today is Dr. Sarah Teichmann, also an expert in all things cell atlases. And this is basically a cell atlas of the developing blood vessel organoid. And what they found was that the mesodermal progenitors ultimately bifurcate into endothelial cells and also mural cells in vitro. And then xenografted blood vessel organoids actually acquire definitive arteriovenous endothelial cell specifications, so they're further subspecified. And they figured out a gene regulatory network, used a single cell genetic perturbation approach to figure out certain transcription factors that are critical for this whole fate specification process in the blood vessel organoid, a role for a protein called MECOM, MECOM, in endothelial cell and mural cell specification in these blood vessel organoids. They looked at the blood vessel organoids and how they're able to generate different organotypic states, again, figuring out the transcription factors that are actually lacking expression in the blood vessel organoids. So that's an illusion to perhaps make them even better. Perhaps you'd be able to introduce those transcription factors exogenously. And that's pretty much what they did. They found that when they induced LEF1 overexpression, it actually improves the specification of brain subtype vasculature, so brain vasculature specificity. And then, they wrap things up with a larger map of the different vascular disease-associated genes to the human brain, the human blood vessel organoid cell states, and then applied this whole model, this whole system, the computational approach and the organoid approach to a human blood vessel organoid model of diabetes. So that was sort of the last thing that they did in the study. So it's an atlas, but it's more than just an atlas. It's the application of the atlas to study, for example, diabetes. And really, this is, like I alluded to at the top, this is one of those really powerful emerging model systems in stem cell biology because everything's got blood vessels connected to it, right? We're all thinking about ways to make our organoids more advanced. And one way to do it is to fuse them with these blood vessel organoids. So you may or may not, you probably will be using these blood vessel organoids in the near future if you're an organoid biologist, just FYI.
Let me add them. These I've been watching this blood vessel organoid field, which is obviously pioneered, I guess, by Joseph Penninger, and he's carrying the baton on, bringing in some collaborators as he's done in the past and applying it toward important insight. The the thing that I'm really waiting for is this angiocrine boss, this organotypic element there, and they get after it a little bit here with that left expression and the brain phenotype. But I really just can't wait for the experience. I think it's just interesting, the idea of a blood vessel organoid, first of all, because like a blood vessel, of all the things in the body that you think of that do not exist like in isolation, like a blood vessel is only there next to a cell, right, to provide it with the sustenance and also these angiocrine factors. So the idea of a blood vessel organoid to me seems incomplete without the experimental applications thereof. And I see that all the work that's being done here, I'm very impressed. But what I've been waiting for is the study that answers this question about the interplay. Does in development specifically, using iPS-derived cells and organoids, assemblage, whatever, to answer the question of how much of the instruction is from the cell, the tissue parenchyma to the endothelium, and how much is from the endothelium back in terms of instructive quality. Because parsing that out, I think, is going to be so critical to really refining our diffs. Because that's, again, there's no cell differentiates in the body at any stage that's not near to endothelium, capillary, whatever, angiocrine factors. So I think that this is something that's important to everybody who cares about cell differentiation, not just for the perfusion function and limits to scale, all that stuff, but really just about how do you get the right cell and how do you get the right configuration of cells to function toward whatever end, modeling or therapy. So that's what I'm waiting to see. Not that this isn't enough. I mean, you look at this; it's OD on the single cell. I mean, dot plots everywhere, heat maps, beautiful, but a lot. But I want to see those critical experiments. Not asking you to do it in this paper, but I'm really looking forward to when we can unpack that.
Yeah, for sure. I mean, it's not a perfect system. Definitely a lot to improve on. I mean, one thing is that a lot of these blood vessel organoid protocols are really intense, really cumbersome. And some they're not always the most reproducible. We were trying to bring some of these into our own lab as well. They're complicated. They're very complicated to perfect, and makes sense why Dr. Penninger's lab is able to really be at the forefront of this. They've been working on this for a long time. But it also goes the other way, right? Of course, like what you're saying, we we see the blood vessel as a conduit, delivery modality of nutrients to adjacent cell types in the developing and the adult body, but it goes the other way too, right? The 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 cell type of association of that blood vessel also matures the blood vessel itself and has an important impact on the functionality of the vessel. And they alluded to it here. So, different cell types could, in the presence of growth factors, immune cells, whatever, that is also important for maturing the blood vessel organoid. So the co-culture is going both ways, and these are that's the power of the system. Is it's not serving in isolation. Of course, there are diseases inherent to the vasculature that you could use these blood vessel organoids for in isolation. But I think what everybody is trying to do is to fuse these with their tissue type of interest, whether it's cardiac organoid, neural organoid, whatever, right? Because it's about maturation, it's about improving the model, right?
Absolutely. And there's nothing more beautiful and horrible than endothelium. And it's that plasticity, as you describe it, that kills you. Once you get it in vitro, it's hard to keep it as is. But I don't know. Is any cell really reproducible, Arun? Maybe every cell, like a snowflake, is an individual. Maybe Dr. Teichmann can weigh in on that, although I'm sure she'll tell me to shut directly up because that is one of the more banal thought experiments I've ever had. But before we get there, I've got a quick message from STEMCELL Technologies. Are you a researcher working with mouse immune cells? You could win up to $4,500 in research tools. If you haven't tried Easy SEP cell isolation yet, join our giveaway for the chance to win an Easy SEP kit and magnet, cell culture reagents, and more. Just fill out the form by May 30th for a chance to win. Visit stemcell.com/giveaway for more information. All right, you guys. Today we have with us a special guest from the world of computational genomics and really spanning all the borders of science and inquiry. Dr. Sarah Teichmann, who is member of the Department of Medicine in the Cambridge Stem Cell Institute, also professor of stem cell medicine. The Teichmann Lab has a long-standing interest in understanding global principles of gene regulation and protein interactions with application to immunology, amongst many other fields. The lab uses state-of-the-art genomics approaches, including multimodal single-cell genomics and spatial genomics, in combination with machine learning methods to advance our knowledge of cells and tissues. Sarah, thank you so much for joining us on the show today.
Thank you for having me.
Yeah, thanks for being here. And we've talked about your work a lot on the show. That's an understatement. I mean, your research has really been at the frontier of single cell biology. You're one of the co-founders of the Human Cell Atlas. I mean, it's really fundamentally changed how we understand the human body, in particular, the complexity of different cell types, right? And I mean, one thing we talk a lot about here on the show is this revelation that there's more plasticity than we thought previously when it comes to cell types. And of course, the work that you're doing has helped uncover that. We've, like I said, covered a number of your studies here on the show. Before we actually dive deeper into the weeds of the single cell atlas and the single cell stories, could you actually just give our listeners a broad overview of what your lab does, kind of in your own words?
So we're a team of about 35 people, and all of us are working together towards understanding the cells and tissues in the human body. We are particularly focused on the immune system. And as you know, that includes the lymphoid tissues, but it also essentially is a distributed system that sort of intermingles with all the tissues in our body. So super fascinating kind of in that sense. And the mechanisms that we discover, we use in vitro systems like artificial thymic organoids to interrogate them in the dish. So we're really interdisciplinary group. We span, from clinician scientists to technical wizards to data science, AML, theory. So it's really very and everything in between. Very interdisciplinary and a lot of fun, basically. And Arun, I guess, or both of you, as you know, we've also kind of had adventures in cardiovascular biology, which again kind of interacts with the immune system in surprising and interesting ways.
Yeah, we're going to come back to that, one of your cardiac stories in a minute. But as you're talking about, and as I've been thinking about, some of the questions I'd pick your brain with leading up to this interview, I've been kind of recognizing how this reminds me a bit, and I was, very young in science, very young period. But it reminds me of the Human Genome Project, that in the sense that like it's this massive undertaking, and it's going to take all hands on deck, mobilization of tremendous resources, and we know it's really important. Like fundamentally, we can feel it; it's obvious, but you don't even know yet how it's going to be applied. We don't even recognize yet how great the scope and the impact of an atlas of all the cell types in the human body. It like literally exceeds our comprehension. But even more awe-inspiring than that, the atlas, this idea of like a static human being and all the cells in the body, we're talking about like encompassing the range of development, aging, health, disease. It's everything. So it's really a huge undertaking, monumental, like the Human Genome Project. And like the Human Genome Project, every incremental advance along the way is kind of a story in itself. All these atlases that have come out are a story in themselves that is really broad in scope. And a couple, I mean, all of your publications really exemplify this, but I want to focus on a couple right now that you kind of alluded to, the Nature and Nature Methods paper this past fall, like literally just months ago, about the fetal and early postnatal thymus, right, atlases, and then the in vitro differentiated T cells. So these two complementary stories that deployed multimodal single cell, spatial, along with these newly developed computational approaches that you just, break off at a pretty rapid clip, it seems, to provide this holistic view, soup to nuts, development, also in vitro, in vivo, everything. Of thymus development. And also, as I said, it's a really important component of how we can leverage the cells in vitro, like therapeutically. Can you elaborate a bit on that specifically, particularly, I guess, about that output there of how these amazing data sets are not just about insight, but can actually be applied scientifically and clinically?
Yeah, I think that's, thank you, Dylan. That's awesome. I completely agree with you that we've only scratched the surface of kind of the implications of, where the human cell atlas is touching on science and medicine. I do want to kind of clarify that the stated aim is sort of to focus on the mapping of the healthy reference human cells and tissues and organs in the first instance, and the disease comparison is kind of an add-on comparator, as it were. Like we simply cannot tackle all 25,000 whatever human disease states. It's just too much, even for the mighty Human Cell Atlas community. But we are very interested in, connecting wherever we can the healthy reference map, to disease, and we're involved in the community, is engaged in a lot of different projects. I would also say, like one more thing, if I may, just on the Human Cell Atlas project, is just to say that you're right, it is reminiscent of the Human Genome Project, and there are a lot of analogies, and it's an interdisciplinary project that provides an underpinning kind of reference data set for the whole community. At the same time, it's a little bit different in terms of sort of the business model for how we've organized the project. We're an open community that everyone is welcome to join and contribute to, and that's different from the way the Human Genome Project was organized, because that was really focused on large centers that had huge, investments in them. Whereas we've had bits of that, but we've also had the big, open community kind of model. And we still do. Everyone is welcome to join the Human Cell Atlas project, and that includes small data sets contributing, or technology, and analysis community, and so on. And then the other point that I also wanted to make is, like you guys have said, the project is really having impacts as we go along. So it's, we're barely halfway there at this stage. But you're right that, we are sort of impacting, in vitro cell and organoid engineering already. We're impacting, disease mechanisms and therapeutics. So it's different from the Human Genome Project that had, one big publication bundle after, I don't know, over a decade in Nature, that had the chromosomal assemblies. Our publication, we've had smaller publication bundles previously, and then we had a large one, like you said, kind of at the end of 2024. And that, has gained a lot of attention, including this podcast, I guess. But it's not the end of the project. That was like, we're at the end of phase one. Let's say we're at the end of the beginning, So there are similarities and differences. But sort of getting towards the thymus, you're absolutely right that we've studied the thymus in using different modalities, single cell RNA plus attack sequencing, so studying the individual cells or nuclei in suspension in terms of their transcriptomic cell states and their epigenetic, chromatin openness kind of phenotype. Also in integrating different highly multiplex molecular single-cell resolution technologies like IBEX with Ron Germain and Andrea Radko, which was the multiplex protein technology, spatial genomics with Visium, 50 micron resolution at that point. And the exciting thing about that paper, well, to me, coming from a data science point of view, was that the way we integrated all of those data sets to reconstruct, a cell atlas of the whole thymus was using a zonation model that we call organ axis, where we have landmarks basically from using a quasi-linear axis from the outside to the inside of the organ. And that allows us to then stitch all of the different modalities onto that non-linear axis and integrate all of the data to make a model of the whole thymus at single cell resolution and the full molecular breadth of the transcriptomics, the epigenetics, and the multiplex protein data. And that is, I think, how we need to be thinking into the future for the Human Cell Atlas. Can we make what I'll call virtual whole organ cell atlases by being clever in terms of the data science approaches that we're using? Because it's too expensive to make a whole cell atlas of an entire human organ or entire human body. But can we get there using imputation, data science methods? And now, of course, we also have deep learning, multimodal AI methods, generative AI, so that we can model whole organs and get towards virtual cell atlas of entire organs. So that's really, to me, that was kind of the beauty of that paper, which was had this organ access software that was instrumental. It was Nadav Yayon and collaborators of ours at Harvard and so on that helped us with the coding and then integrate all of the data onto that single framework. It's a model. It's not that we measured the whole three-dimensional thymus at single cell resolution in one shot, but with the model, we're able to reconstruct all the layers of the onion, so to speak, and place the cells, in a kind of reference framework for the whole organ. And that's important and powerful because it then allowed us to trace where the T cells are going on their journey when they enter the thymus as a hematopoietic stem cell, as an early thymic progenitor, and then they go on their maturation journey around the outside, the cortex, and then through the corticomedullary junction into the medulla. And it's that 3D model that allows you to trace that journey and track the different tissue microenvironments that the cell is seeing, so to speak, as it matures. So that I think is kind of an exciting era that we're in, so to speak. The thymus is a small organ, right? It's a couple of centimeters and sitting next to the heart, not as well known as the cardiovascular system, maybe because it was only discovered in the 1960s as an organ with an immunological function. But nevertheless, it's vital to life because kids who are born without a thymus die unless they're treated with stem cell or or thymic transplant. So it's small but mighty.
Yeah, you talk about the importance of models, and I think we've all heard that phrase that all models are wrong, but some models are useful, right? And I think this is one of those situations where this is a very, very useful model for the greater community and beyond the just thymus community, but just in general. And thank you for looking to the future for a couple minutes there. I think that was really interesting, and thinking about ways that we can incorporate machine learning on all these next gen AI approaches into integrating with some of these data sets. But I was actually hoping you could also take a look back. I mean, this is a massive undertaking, the Human Cell Atlas Consortium. It's, like you said, numerous investigators across the globe helping us to unravel cell biology at the single cell level. I mean, the consortium itself actually just started under 10 years ago, if you hard to believe, co-founded by yourself and also Aviv Regev, who's at the Broad Institute. Could you take us back before those 10 years and just talk a little bit about how this came about and how you actually managed to attract all these brilliant minds from around the world to work on this singular topic?
Yeah, no, it's fun to look back and kind of reminisce. I mean, the I would say birth of the Human Cell Atlas is closely linked to developments in technology, and that's single cell genomics and spatial genomics, and then the data science kind of to stitch the two together in a sense. And as I'm sure you know, single cell genomics sort of, to me, was very exciting to read the first paper that profiled the transcriptomes of a handful of cells. And it was the group basically just down the road here in Cambridge at the Gurdon Institute with Azim Sourani, whose group studies epigenetics and primordial germ cell development. And they had handpicked, 4 or 5 cells and then profiled the transcriptomes of those cells from the mouse blastocyst. And then later on, so to me, that was kind of a revelation, because if you take that and logically sort of extrapolate it up, you can profile an entire organism, a worm, a fly, and ultimately someday human, cells and tissues and all the whole human body. That's not what they were trying to do, because they were focused on their challenge of there being only very, very, very tiny numbers of developing primordial germ cells, and that was motivating them to get to a single cell resolution, which is what they achieved. But for me, in terms of coming from computation, data science, and also the biological cell compartment that we were studying, which was T cells, the way I was thinking was, can we dissect decode the different cell states in an unbiased way in a heterogeneous sample? And the more cells, the better. And what happened, of course, very quickly was that, both here and in Cambridge and elsewhere around the world, and particularly in Sweden, with Sten Linnarsson and Richard Sandberg, but also in Israel with Itai Yanai, and in the US, and in papers, a lot of different papers came out with slightly different protocols, different approaches, different scales than in single cell genomics, and that was really exciting. Azim, was really key for us because he helped us access the FluidMC1, which was the chip-based microfluidics instrument, and that's kind of how we succeeded in catching T cells, single T cells originally. There was also a doublet problem in the chambers, but where there were single cells and doublets that were captured in these microfluidic chips, and I actually kept that chip-based system, which isn't really used anymore nowadays, but I've sort of got it on my desk. And so that was really, really incredible. And at the same time, of course, there were more and more imaging-based and sequencing-based multiplex spatial, I'll call them genomics or spatial biology methods that were developing kind of in tandem. The in situ sequencing methods from Mats Nielsen were coming. The kind of related but slightly different Murfish method from Xiaowei Shang, the spatial transcriptomics from Joachim Lundeberg, all of those, that sort of area was developing then shortly afterwards. And so if you take those two developments together, you essentially have a human cell atlas, right, from a data generation point of view. And I guess that then made it clear. So backtracking again, telling the story from the Cambridge point of view, which is the point of view that I know best. I was at that time in the MRC Laboratory for Molecular Biology, which is on this campus, the biomedical campus in Cambridge, where the hospital is and so on, where the clinical school is. And there's another campus, which is sort of 8 miles south in a village, which is the Wellcome Genome Campus that has the European Bioinformatics Institute and the Sanger Institute. So it's got sort of computation and genomics out there. And, while there really wasn't a super lot of interest or kind of appreciation of these single cell genomics technologies on this campus at that time, although I should say that Bertie Gutkins, who's the director here, was, interested in single cell qPCR, single cell gene expression, and so on. Even then, out on the genome campus, there was really a community that was starting to form, and I was very fortunate in the sense that I was recruited out there by both the EBI and the Sanger, and I was the only, the first and today only faculty who was kind of co-recruited into the two institutes. And at the same time, the two institute directors, Janet Thornton, who was also my postdoc advisor, kind of in a different life, kind of in a bygone era, and the Sanger kind of committed to the so-called Sanger EBI Single Cell Genomics Center. And when we founded that, there were 5 of us who founded that, and that was, all kind of evolving during 2012. And then I had a maternity leave. My second daughter was born in August 2012, and so I joined in February 2013. And that meant that there was 5 of us: John Marioni, Chris Ponting, Wolf Reich, myself, and then a staff member called Harold Swardlow, who were all working together with a budget of about 4 million or so from across the two institutes for tech dev in single cell genomics. And we called it the Sanger EBI Single Cell Genomics Center. And that meant that we were able to sort of run together because we were all struggling or tackling challenges in cell isolation, nucleic acid amplification, sequencing, data analysis, blah, 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 blah. And it became a sort of like the cool kids kind of club, if I may say. I'm almost 50, but
You may absolutely say you are cool as ice, Sarah.
And then, there were a lot of other people who were interested in joining, and we had seminars every few weeks. And on the data science side, John Marioni and I had adjacent labs, and then Oliver Stegle was next to us as well. And so we had a really good community, and there was a great kind of activity around on the experimental side and multiplexing, and multimodal technologies with the genome and transcriptome sequencing and so on. So yeah, that was a really fun time. And that, what was very clear was that if those, at that time, somewhat arduous and, was a hard slog developing all of these technologies, but if they would scale, then, there would be a human genome project. And then in 2014, Aviv pitched to NHGRI the term human cell atlas as part of a workshop. It wasn't funded, but it was, the pitch was out there. And once I was then appointed into the Sanger head of cellular genetics departmental kind of position, I reached out to Aviv and said, hey, by that time it was clear. So that was late 2015, early 2016. By that time it was clear that, this wasn't a completely outrageous, ridiculous idea. This could actually work because, of course, we got the microfluidics technologies came online then. And I should declare I'm a non-executive director of 10x Genomics, but those droplet robots really changed things because they meant that you could do experiments very robustly and reproducibly, and with a kitted kind of commercial solution. And that then, that also kind of made our life a lot easier and more fun, kind of in terms of rapid data generation. So we sort of kicked off the project around that time when everything was taking off with and Aviv and I, we started planning a meeting then. We had weekly calls, it was pretty intense, actually. And I mean, the good news was that, we got support from funding agencies. So Wellcome gave us a 50,000 award for the first meeting in London in October 2016. And the other good news is, Aviv and I were very aligned, and that very rapidly other people, the community kind of banded together and came together to form the organizing committee of the Human Cell Atlas at the first meeting. That was about, over 20 people from all different parts of the world, different biological backgrounds. There was the Swedish colleagues that I mentioned, Hans Clavers, with the organoids, could see the point because of, what Dalen was saying about in vitro and vivo comparisons. He obviously comes from the in vitro side and really wanted to know, how do my model systems compare to the human gut, at that quantitative single cell molecular resolution. The people, as you said, also the Human Genome Project, there were people who knew about that legacy and how powerful and influential it's been, and wanted to then take their experience and skills, into the Human Cell Atlas. Partha Majumdar is one example, who's from India, Calcutta, who'd been part of the Human Genome sort of days and joined Human Cell Atlas. So it was a really, yeah, I mean, John Marioni, whom I've mentioned, lots of people, and that was, very, very exciting. I want to mention the next Human Cell Atlas general meeting is in Singapore in early June. Be there or be square.
Yeah, you don't want to be called a square by Mrs. Cool over here. Yeah, well, I mean, this what comes across is the strength of the idea and the utility, right? Really carried it off in large part. In addition, I don't want to understate or not give credit to that very heavy lift that you guys all did in the genesis there. But what's weird about hearing about it is that we're talking about this thing like it's something that happened, like Watson and Crick. Like this is ongoing. This just happened.
Hindsight is always 2020, Dale.
Oh, for sure.
Hindsight is always 2020.
For sure, but I would say
that it was so obvious and influential, but actually it was not obvious.
Oh, for sure. No,
Okay.
I think it took a lot of courage for sure. It took a lot of courage, and like you said, the risk and getting the commitment and the buy-in. It was all very, very.
There were a lot of critics.
I can imagine.
I'm not going to name them, but there were a lot of critics. There were also people who just didn't get it and were sort of.
Well, I can say, I mean, as someone who I remember hearing about the first breakthroughs, I think what the challenge was, because I remember you would do like the laser capture. That was I don't know if you remember it, but the laser capture, you take a cell and you do you did the amplification with the Arcturus Pico Pure 1-cell input, and the results would be spotty. So I think what a lot of people who didn't have the faith in I think the capability of the data scientists to really delve in, but also the chemistry. I think the chemistry didn't exist; it had to be created. And again, a testament to you and the team and all of the inventors who helped to carry this off. And as I started to say, I think the amazing thing to me is that I don't want to say we're building the plane mid-flight, not we, but you, because that's not exactly right. Like you said, you're making the insight, your innovations, like we're learning a lot from this and applying it toward clinical and whatever, scientific inquiry and insight already. But what's interesting to me is that the tech is still evolving so rapidly, right? You to look at just like spatial as an example. In recent years, I know you were an early adopter a couple of years ago with like the cardiac niche thing, and that really illustrated the power, I think, and used it in a lot of clever ways with the drug prediction and all that stuff. But like the key there for me was the notion of the niche, right? You can dissociate and see the single cells on a single resolution level, but the see in the niche is so key. And you mentioned the 50 micron resolution at the point of the thymus story. And since then, now they have the high D, right? Which is, I mean, you're probably in on those meetings, which seems to me almost impossible. But hey, if you tell me it's true, I'll believe it, Sarah. But the tech also and the computational approaches that integrate it, as you elegantly described in the thymus story, like the progress seems to be advancing and accelerating. So I ask you, is it matter now of just like deploying the tech as is across the scale of the healthy reference data set of the Human Cell Atlas, as you mentioned, or are there still some hurdles? Like as someone who really is on the ground, what are some, or are there any hurdles left technically to overcome? Resolution being perhaps one of them. Depth, I don't know. You probably know better than anybody.
Yeah, I'm on the ground. I'm in the trenches. I'm at the coal face. Yeah, so I'd say it's an exciting time, like you're saying, because we are getting to that single cell resolution and very, very high plexity at the molecular level, and that is incredibly powerful for us to learn about cells and tissues. I guess where I see the kind of future is, yes, we just have to generate data for sure, and there are various challenges, in tissue handling, preservation, and scaling, and so on and so forth. There's also, that the thing that I was mentioning before is how do we make whole organ cell atlases? And the way we did it for the thymus was really bootstrapping by modeling. And as Arun said, you said something like models are
all models are wrong, but some are useful.
all models are wrong, some are useful. So, can we get more, more data and less modeling, or more useful models to make whole organ cell atlases? And do we do that by simply, consecutive sectioning, like thin sections, and then integrating them into 3D volumes? Do we do it by developing more depth in the imaging and sequencing technologies in the z-axis? Do we do it by more deep learning and generative AI reconstructions that are better models, than organ axis? And so how do we achieve that? There are a lot of different strategies. Which one will win will probably also depend on, the particular organ and tissue and the symmetry constraints and so on and so forth. The other kind of so that's kind of how I see things going in terms of whole organ cell atlases. I'll just use that word, or virtual whole organs. Say. And what we want for the human cell atlas is ultimately a model of the human body, right? That we can navigate and we can look at all our cardiac and vascular and so on cells in 3D. The other or the other component of that is the multimodal aspect. So we've talked about, transcriptome and epigenetics, but what, and multiplex protein. And can we model them all? There are different aspects of the cell's phenotype, the cell's shape, the morphology is what we're also getting now from those technologies. Like you mentioned, Visium HD, you always have, also in Xenium, you always have the H&E, the stain, whatever stain that you have. So you see the shape of the cells, or, cardiac myofibers, Purkinje fibers, whatever, you see the shape. Can we relate the shape to the molecular content, protein, RNA, epigenome, and so on? That's what we want to be doing. And that's, there are technical and computational, or technical experimental and technical computational challenges to get there. But that's what I think what we all want in order to get the most comprehensive description of each cell that we can. So it's what we've called, we had like our group wrote a sort of opinion article in PLOS Biology where we called molecular to morphological, reconstruction of cells and tissues. So you want both the morphology, in other words, the shape, but you also want the molecular profile, the molecular resolution, and obviously at full plexity, or as broad plexity as you can.
Yeah, I mean, you talked a little while back about the unique nature of your training, right? And I think that lends into how you've become this leader in this, unique field that's at the intersection of computational biology and quote wet lab biology. I mean, this is like you said, it wasn't easy to get into this field and get started in that approach, and not everybody was a believer, 10 plus years ago. But now it's almost like the norm to integrate single cell biology with wet lab biology, and pretty much every institute across the world. It's become so commonplace now, right? And because this is really a trainee oriented show in a lot of ways, we were hoping you could talk a little bit about that style, and perhaps how the next generation of trainees in biomedical research should integrate computational biology with wet lab biology. For me and Daylon, this wasn't a reality during our training, and I'm sure for you as well. But now it's so accessible to become a computational biologist in some capacity, whether it's learning basic coding or something like that. So, what advice do you have for young scientists, trainees that are looking to work at this intersection of disciplines? I mean, it's that age-old question, right? Do you specialize? Do you have to specialize? Or is it more important to become a generalist these days?
Yeah, it's a great question. And, like you said, nowadays, yeah, all the trainees, to them, it's like a matter of course that, we do our single cell experiment, analyze the data and so on. And that's, it's awesome. I mean, it's really awesome how things have evolved, and, and that I obviously feel somewhat vindicated, because as I said, there was such a, I was in such a community that was not a believer in that, I'll say, when I started this work. That company Fluidigm, when I showed you those chips, they came to give a talk at the LMB, and there were 4 attendees at the talk. Which was myself, my postdoc, and then 2 immunology PIs, one of whom's left. So it wasn't like there was, 800 people all realized that this is the future. It was very, very far from that. On the Genome Campus, it was a bit different. I mean, I would say, for us, or I guess for me, the data science, the era of data science, and the importance of, computational theoretical approaches. Now, it's an amazing era because of all the deep learning and generative AI. That I think it's safe to say that will continue the rise of computational technologies in science. That will continue into the future, and I think people really need to engage with that now. You just can't ignore it. And when I started my PhD in the mid-90s at the LMB, I would say that was not the prevalent kind of view, but in any way, shape, or form. In my training, I was very, very fortunate, because I studied natural sciences at Cambridge, and that course still today kind of allows you to choose different, so you don't have to commit to physics or chemistry or molecular biology, but you enroll in like this general course, and then you can choose different courses within it. And so it meant that I started off in physical sciences, so Master of Physical Sciences, physics, chemistry, and then I took like an introductory course, and it was called Biology of Cells, and then gradually I sort of moved over into biochemistry. But it meant that there was some scope for interdisciplinary training within the university course, and that was an amazing opportunity, I would say, because it sort of set the stage then for doing like my bachelor's degree project in NMR spectroscopy, which was purely computational project, and then the PhD, which was purely computational in, I would say, sort of genomics and proteome and 3D protein structure analysis with SARS-CoV-2. So that was kind of, I would say, the natural science course enabled that. Nowadays, I'd say, I'm biased, but I'd say really, computer science, data science, AML, like more statistics maybe than what we had, kind of more conventional maths, like would is incredibly important. But at the same time, I hesitate to sort of be prescriptive, because everyone finds their niche in a way. And it, even if it's not data science, there's people who are amazing at connecting surgery with cell atlasing, or, there are all kinds of ways that, that you can contribute to science. It doesn't have to be through that in an interdisciplinary way. So I think the key really is that there's a lot of creativity that comes out of interdisciplinarity in my experience, and in my my personal kind of observations over my scientific career. And I guess that's more the key, as opposed to sort of being prescriptive about what that specifically needs to be, if that makes sense.
Yeah, I mean, if it's what's fascinating about biology, it's just there's more. You'd think after all this time we would have figured it all out, right? But obviously not. There's an infinite amount of discovery, it seems, out there. And what you're talking about, to me, it strikes me as like, yeah, we're going to get to a point where you need these computer learning, machine learning approaches to like see into the data, because it's more than any human mind can really wrap itself around. And you may have the AI like showing you things that you couldn't see by yourself. You know, you hear about AI doing this deep math that humans can't even understand. And that may be the case, but at the end of the day, you're always going to need maybe someone, notwithstanding some robot overlords in the future, you're going to need a human being to like test it, right? And you're going to need the creativity to be interested in the question. And I think that
And
you
even
know,
identify the question.
right, identify of all, that's what AI, you know, you search anything on Google these days and you get a million answers. But like the reality is what's the answer that matters? And I want to believe, you know, I have kids who are approaching adulthood, and I want to believe that there's still a place and they'll still have an opportunity to contribute to society. And I have to believe that the way they're going to do that is by both incorporating the tools that are amazingly at their disposal, but also, you know, maintaining, you know, an open mind and maintaining their creativity and believing in their ability to contribute. I think that's the scariest thing about AI is that there's this doom saying it's like, do I even matter anymore? What's
No,
the point?
that's what people say over and over again. You know, I was at a conference in Edinburgh last week, and it was exactly the same thing. But that's not how I look at it.
Hmm.
So to me, it's just a very fun, powerful tool.
Mm-hmm.
You know, just like that microfluidic chip, you know, that I showed. It's another kind of tool that we can deploy, and it will just accelerate things for us, and it's making our life easier, and it will help with coding. That's what all the trainees are just using, you know, generative AI for coding. I mean, that's amazing to me because we spend like so much time, you know, writing our own code, whereas now it's all just, yeah, do this, do that, do that. Thank you, you know. But you still need to tell the AI what to, or need to ask the questions, tell it what to do, make the choices. So I don't have that sort of doom and gloom kind of view. And I'm not concerned about my daughters, you know, one of whom is going to be an adult within the next year, you know. So I see exactly where you're coming from, but to me, it's just another kind of tool. Do you see what I mean?
Oh man, I'd love to hear that from you because I think that you qualify as an expert, Sarah. But that does put weirdly hearing you say that I guess you're kind of implying that there's always going to be limitations on these tools that you yourself are creating. So that to me suggests that there's still a role for us human beings. So thank you for sharing that. And before we let you go, I have a couple of peripheral questions. The first being, if you could answer any single scientific question, which maybe you won't have to now that the AI's here, just kidding. If you could answer any scientific question, regardless of your expertise, what would that be?
Well, what I'd love to know is, you know, when we finish the human cell atlas, how many cell types are there in the human body?
Hmm.
You know, and that's something that we are still a ways from knowing. Wikipedia says 200, you know, the mouse brain has 5,000 that have been described. So knowing really what are all the cell types in our body, I think that will be transformational and something I'd love to have the answer to before I retire.
Well, you've got a lot left to contribute, so get to it, and I'm sure you'll get there. Although there is an argument for isn't it like a snowflake? Every cell is different. I think that's a little bit of an oversimplification. Finally, what is the biggest misconception about science that you would like to resolve?
A misconception that I think we have is that scientific discoveries are made by individual geniuses.
Hmm.
In reality, I think every single thing you know, every single solution has been developed, you know, by amazing individual people, but it's always influenced either working together with or influenced by interactions with other people. And, you know, we're, as a scientific community, as a human cell community, but as a scientific community, we're one team in the end. And, you know, we can all learn from each other and work together to advance science and medicine, I think, in the end, for the benefit of humanity.
Well, I love the symmetry, if nothing else, of that answer because the first two words you said in this interview, besides thanks for having me, were my team. And I really appreciate that you led with that because I think it underscores both, the many brilliant minds that contribute not only to your work, but this whole endeavor of scientific inquiry. But also, it speaks to what a great leader you are in acknowledging the credit as you began and ended with that. And I think it's a testament to also the amazing consortium that you built, not only in your own lab, but in forming this initiative with the Human Cell Atlas. So thanks for all of your work, and particularly thanks for giving us a bit of a kind of origin story there, which for me really illuminated what a tough fight it was. And I'm glad you shared that with our audience, because we take this stuff for granted, like, brilliant ideas just stick. But like, the truth is, you need people to really showcase them and to fight for them, and to make people, skeptics like myself, who don't believe in the chemistry maybe, or the laser capture, whatever it is, to make us really believe. And I talk about a 180. I look at my single cell data every day. It's just an infinite font of discovery and insight. So thank you for your contributions, and thank you for sharing your story.
Thank you for having me. It was a lot of fun. Appreciate it, and enjoy the rest of the day.
All right, you guys, that brings us to the end of this show. What a great conversation with Sarah. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on X at StemCellPodcast, Blue Sky at StemCellPodcast.com, or via email at info@StemCellPodcast.com with feedback or to suggest guests.